Good morning, good afternoon, and good evening, everyone. On behalf of Janelia and 7 other sister institutes from around the world, I want to welcome you to Life Science Across the Globe. My name is Janine Stevens, and I am one of the Janelia organizers of this series. And today's event is focused on plant biology and is being hosted by the University of Buenos Aires and CONICET in Argentina. We will have 3 short talks followed by a moderated discussion and then audience Q&A. Please go ahead and write your questions in the Q&A box at any time, and we will ask them during the Q&A period. We will end with a Meet the Panelists session where we invite students, postdocs, and other trainees to stay on the call and ask questions to our panelists and hear from them on various career development related topics. I also encourage all of you to please take a moment and complete the very brief survey that we will link in the chat box because your feedback is really important to us. So now it is my pleasure to turn it over to Professor Alberto Kornblit, Director of the Institute for Physiology, Molecular Biology, and Neurosciences at the University of Buenos Aires and CONICET. Alberto.
Thank you, Janine. Good morning, good afternoon, and good evening. My name is Alberto Kornblit, and as Janine mentioned, I am a professor at the University of Buenos Aires and investigator of the National Research Council of Argentina. Both UBA and CONICET are the hosts of today's seminar, and I am extremely pleased to introduce our moderator, Professor Jonathan Jones. Professor Jones is a group leader at the Sainsbury Laboratory in Norwich, UK, and professor at the University of East Anglia. He has made seminal contributions to understanding molecular mechanisms of plant resistance to pathogens. He is a member of the Royal Society and a foreign member of the US National Academy of Sciences as the 3 renowned international scientists who will join us today. Without further ado, I pass the floor to Jonathan.
Thank you very much indeed. It's a great pleasure to be able to host this HHMI event, Life Science Across the Globe, on plant science, on this theme of mechanisms meeting biodiversity. Plants are so important. Every day they achieve that miracle of using energy from the sun to convert CO2 into sugar so that they can grow in hugely diverse environments, enormous areas, and despite multiple stresses they have to cope with. And they do so by adapting fundamental biological mechanisms that are common to all organisms. They have to transduce information from their environment into appropriate responses. So they have to be able to perceive and respond to their environments. They also have to reproduce and create viable progeny to maintain each species over generations. And we'll see these themes picked up by today's speakers. So without further ado, I'd like to introduce our first speaker, Sandra Díaz, is a leading world authority on plant biodiversity and on ecosystems, plant functional traits and platforms of functional specialization. She combines her plant ecology studies with interdisciplinary work on how societies value and reconfigure biological communities and ecosystems. And that's a theme she'll be pursuing today. Sandra, look forward to your talk.
Thank you very much. Good morning, everyone. I am a macroecologist. I hope you can hear me well. I really feel very honored to be a part of this series of webinars. One of the most remarkable things about plant life on Earth is its astonishing diversity of form and function. As well as being a continuous source of inspiration, this poses a formidable challenge. If we are to understand and manage biodiversity and ecosystems, we can't deal with each species as a separate case. We need to identify some general principles underpinning this exquisite complexity. So, we set out to describe essential axes of specialization at the level of the whole plant, at the global scale, and considering all 3 major functions in a plant's Darwinian struggle for existence: growth, survival, and reproduction. In order to make this operational, we needed to find a minimum set of plant traits that together would encapsulate all these functions and at the same time would be available for a very large number of species worldwide. So we finally settled for plant height, seed or spore mass, leaf mass per area, leaf nitrogen content, leaf lamina size, and stem specific density. Thanks to the TRI global communal database of plant traits, we managed to build this dataset. It contains a very wide variety of vascular plants, and it contains also the most extreme values of these fundamental traits known to science. And it includes over 40,000 vascular plants. So still to this day, and to my knowledge, this is the largest data set in the world in terms of species mean values and geographical and botanical breadth. And what we wanted to do with this is to find out how the multidimensional trait space occupied by living vascular plants looked like. What was its outer shape, and how were species distributing within this volume? So if we imagine a 6-dimensional volume, each dimension corresponding to one trait, if every combination of trait values were possible and equally popular, then we would observe a hyperdimensional, quasi-spherical, serenely homogeneous cloud or blob. So we did build the observed, the empirical 6-dimensional trait space. And you can see here some 3-dimensional projections of it. And then we tested this observed hypervolume against a group of formal null models. And in a word, we found that the observed phenospace of vascular plants is definitely not a quasi-spherical homogeneous cloud. There are strong constraints to what combinations of traits are possible, or at least likely. The multidimensional trait space is rather flat and constrained, like the galactic disk of the Milky Way. Or using a more domestic metaphor, if you like a flat bread. The trait space actually occupied is so constrained that about 3/4 of the whole variation is captured by a single plane. This plane, which we name the global spectrum of plant form and function. This plane has two major axes of variation. One axis is in this direction, reflects the size of whole plants and their leaves and seeds, goes from small plants with small leaves and tiny seeds all the way to very tall plants with large seeds and large leaves, from that width to Brazil nut. The second major dimension, this one, is a leaf resource economics axis, from conservative, slow-living leaves like those of holly, or even more conservative, the Australian IGCAST, all the way to acquisitive, tender, short-lived leaves like those of basil. This is the first global picture of essential functional diversity of vascular plants. Now, let me show you now some illustrations of the kind of plants located at the fringes of this plane for your reference. Here, in this area, we have the Brazil nut, pines, junipers, and monkey puzzles here, IGCAST, heathers, large floating and tiny submerged aquatic plants, the tender but toxic devil's snares and henbits, And because many of you work in genetics and molecular biology, I thought you would be interested in knowing where is Arabidopsis thaliana. It's here, as you can see, in a remote, sparsely populated corner of the functional galaxy. So this spectrum of form and function can be used as a backdrop, as a navigation chart on which one can place any vascular plant species or community on Earth, and can also be used to understand the selective and evolutionary trajectories of these plant entities in the past and in the face of ongoing and future environmental change. And the global spectrum is actually being used and furthered in a number of projects worldwide. The first question these new projects address is, are these two dimensions enough? The simplicity of the global spectrum is actually its strength and beauty rather than a weakness, because you can capture with it the essential design of any plant entity with only two quantitative continuous coordinates based on well-established theory and applicable to the vast majority of vascular plants. But this spectrum doesn't encapsulate everything that is important to a plant. This pattern should be seen as a minimal set in the mathematical sense. Any functional description of what the plant does should consider them, but it doesn't mean you are going to be able to explain everything with these patterns. For example, only last week, we published a new paper on how fine root traits map onto the global spectrum of plant form and function. And we found that fine roots actually add some non-redundant, meaningful information to the global trait space of plants. Here you have the global spectrum as I just described, and this is dimensions 3 and 4 of PCA, which are all fine root traits. But this and all the analysis I have seen so far, when they incorporate new traits, the extra dimensionality doesn't override the first plane. So going back to the flatbread analogy, the trait space occupied by vascular plants is not totally and homogeneously flat like a Mexican tortilla. Rather, it looks more like a chapati or pita bread with varying thickness, with lumpier and flatter places, but the hypervolume is still predominantly flat, dominated by size and leaf resource economics. Another important point, which I thought you would be interested in, is the fact that this is a plane only in the mathematical sense. But if you add a probability density to it, which in this map has been represented by the colors, you actually get a topography, a landscape, with red hot peaks, which I call functional hotspots, with many species that are very similar in terms of their functional trait combinations, highly redundant if you wish, and also other much paler, colder valleys, like this, which I call cold spots. These valleys represent trait combinations that I either not observed or do exist, but are very uncommon. Indeed, we were expecting constraints to the multidimensional space, but we were not expecting this striking topography. And this topography is not the result of some odd splurge in speciation at some point of evolution. As you can see, many distant families and orders are located in these hotspots, indicating that these are successful solutions found repeatedly over the evolutionary history of vascular plants. Now, on the other hand, some perfectly viable combinations of traits from the biomechanical and physiological point of view are very rare or definitely don't exist. Presumably because they are eliminated by or are under strong negative selective pressure from natural selection. Now, the question we and other colleagues asked next was, can this topography of the functional landscape of vascular plants change over time? Can the peaks and the valleys shift? And do we humans have a role in it? You are probably all well aware of the dramatic extinction rates of many organisms at the moment as a result of anthropogenic causes. And plants are not exception. But these general numbers obscure the fact that different kinds of plants appear to be threatened to very different degrees and in very different ways. And these taxa happen to be placed in very different areas of the global spectrum. So we use the sampled red list, which is a non-biased version of the red list of the International Union for the Conservation of Nature, and mapped these species from the sample list onto the global spectrum. This still work in progress, but quite roughly, least concerned species seem to be spread everywhere in the spectrum. Near threatened species are more restricted, and threatened species are disproportionately located on the right side of the plane, and especially the southeast corner, so to speak. And this corner happens to be an area in trade space that might have been a hotspot in the distant past, but no longer is one. So here you get low redundancy as well as disproportionate threat. So this area here is a blinking red traffic light in the functional space of vascular plants. But human pressure is not only about thinning already thin areas of the global plant phenospace. We found something interesting in this particular area of the spectrum, which is a really acquisitive, nitrogen-rich, live fast, die young neighborhood. This is the area in which are located most of the species strongly related with us, either because they are our crops, peridomestic weeds, or they are sources of poisons, or recreative or medicinal or traditional drugs for decades, centuries, millennia. You don't get a species hotspot here because in the wider picture of the world's flora, there are not that many species in that particular corner. But if you look at the level of varieties, there is a huge concentration of them. And they are disproportionately important to us and to a very large degree created by us. So this is a natural functional cold spot in phenospace, in which we have been trying to build a warmer spot for millennia. But our deliberate sculpting of plant form and function, remarkable as it is, has involved only a tiny fraction of the whole phenospace of plants. So, wrapping up, the phenospace of vascular plants is remarkably constrained and lumpy. The variation is largely concentrated into a plane, the global spectrum of plant form and function, defined by plant and organ size on the one hand and leaf tissue quality on the other. This plane doesn't capture everything that is important about plants. It has to be seen as a minimal set. Other dimensions are likely to enrich the spectrum, but so far appear much less important. The spectrum is strongly granular, with strong conversions towards a relatively small set of successful trait combinations. And finally, the present model of human appropriation of nature has clearly been shifting and is likely to continue shifting the topography of the global spectrum. To finish, I would like to share with you two wider informal corollaries. First, despite their amazing superficial variety, most vascular plants share similar combinations of these 6 essential traits. And second, despite our amazing capacity to manipulate plant form and function, our deliberate sculpting of phenospace has been focused on a tiny corner of the global spectrum. And at the same time, we have been causing highly biased collateral damage in other regions of trait space. And I would like to thank all the colleagues that participated in this work. Thank you very much.
Sure how we applaud, but let's applaud. That's absolutely fascinating account of bringing science to documenting the extraordinary diversity of plants that we all know and love. And so, without further ado, we're going to move on to our second speaker, Caroline Dean. She's a leading authority on plant responses to temperature and on plant gene regulation by both genetic and epigenetic mechanisms. And she's going to be describing how plants monitor both acute and long-term temperature signals to induce a switch that a particular gene, the flowering locus C gene, to silence it and thus allow flowering to occur. Caroline.
Thank you, and good morning, good afternoon, and good evening to everybody. We've heard from Sandra about the wonderful diversity of plants, but one common thing of plants is how they use their environment for decision-making on a daily basis to regulate their metabolism and on a longer basis to regulate their timing of development. And a picture here shows a classic timing of development picture, a seasonal transition of a tree through the seasons in a kind of typical Northern Europe environment. And the question is, how are plants actually perceiving these very complex, often seasonal cues and using them in developmental timing? Now, the seasonal cues, the main ones in this part of the world, would be photoperiod and temperature. But of course, photoperiod is the same in autumn and spring. So spring, which is the main season for a lot of drivers for developmental decisions in plants, is always a period following prolonged cold. That's what the plants use to judge when winter has passed and spring has arrived. Now, for a long time, people thought, well, that's a long period of cold temperature. That's how plants are registering cold. But if you look at what the plants are actually registering, and here's a plant sitting in a field in Sweden with a temperature monitor next door to it, looking at the hourly temperature changes, you see the fluctuations the plants are experiencing are enormous. a daily temperature fluctuation is greater often than a whole seasonal fluctuation. So the question is, how are plants coping with these noisy signals? And how are they distinguishing today's signal from yesterday's, from last week's? What's the memory and the memory mechanisms of these prior exposure, so they can look at and measure seasonal changes as opposed to daily changes? And it was those questions that got me into this whole topic of vernalization, which I'll talk about today. When I set up the lab, we knew a lot about vernalization in crop plants. Vernalization is the acceleration of flowering by this prolonged period of winter. It aligns flowering with spring conditions. Very important for many crop plants, wheat, many other cereals, brassicas. But fortunately for us, it was also very important in the reference plant Arabidopsis thaliana. Now this panel here shows the typical characteristics of vernalization on Arabidopsis in this case. If you germinate the plants and grow them with no cold exposure, they flower very late, they keep remaining vegetative. But as you give them weeks of cold before you put them in your nice warm chamber, here case 3 or 6 weeks of cold, you progressively accelerate flowering. And that showed these classic true characteristics for vernalization in all plants. It was relatively slow, it was quantitative. When you took a cutting from this plant, it would remember it had had vernalization and flower at the appropriately according time. And it was therefore mitotically stable, and it was reset every generation. Now, there had been a lot of work on models of what might be accumulating through this temperature time series. Crop models are used to predict flowering time of crops in the field, in fact, for vernalization, but no molecular understanding. And through a combination of many different groups, our understanding now is that certainly for Arabidopsis and related brassicas, a key player in this whole process is this gene that Jonathan mentioned, flowering locus C, FLC. You just need to think of this gene as making a protein that's a flowering brake. If it's on in the plant stays vegetative. So as the plants germinate in autumn, FLC transcription is very high, this brake is made, flowering stays off. But as the plants experience winter, slowly but surely through these fluctuating cold temperatures, FLC expression is reduced. But then upon reduction, it is epigenetically maintained at that silent state, even after the plants come back into the warm conditions of spring. And in those conditions now, because the brake has been released, taken away, the photoperiod, the warm conditions, all of the other things required for flowering can actually stimulate flowering. Flowering occurs, but of course, to make sure every generation needs to overwinter during seed development, FLC expression is reset. Now, through the forward genetic analysis, it became clear that this silencing and epigenetic maintenance involved chromatin regulation. And I draw these nucleosomes schematically here, color-coded with a traffic light system. So red is for silent chromatin, green for active chromatin. And these come, of course, with certain modifications that now are well characterized in many systems. For vernalization, it's chiefly this methylation on histone 3 tail at lysine 27 trimethylation. This is our red traffic light silent chromatin state, typical of what's been called polycomb silencing, initially discovered in Drosophila, very important in mammals and also in plants. So we got into a chromatin silencing mechanism. So one of the first things we asked was, how is this process of vernalization quantitative? So we looked over the gene, and the schematics on the bottom here. This is the exon structure of the gene. Here's this arrow shows the transcription start site. And this little red bar here shows a region I'm going to call the nucleation region, because that's where we see in the cold, if you give the plants increasing cold, you see progressive increases of this K27 trimethylation by chromatin IP at this nucleation region. And it's only after the cold, as the plants grow out in the spring, that K27 trimethylation spreads across the body of the gene. So we'll refer to nucleation and spreading. But of course, this picture gives a slightly misleading picture. It looks like there's quantitative accumulation at K27 trimethylation at each gene, but it's not that at all. What we found was actually this was a switch, and it was a population average effect that we were looking at in these molecular experiments. If you looked at individual cells, and I'm showing you here Arabidopsis roots expressing an FLC venous fusion, you can see that I show you roots because actually they're easy to image. They're also beautiful systems because most of the cell division is in the meristem down here. And these files of cells, because of anticlinal divisions, are developmental lineages. So it's a very interesting and useful tool to actually look at the epigenetics of expression states. But vernalization is a whole plant feature. All the tissues of the plant vernalize. FLC will go down in all of them, certainly in the meristematic cells where it's mainly expressed. So in plants with very little cold, you see FLC in all the nuclei. But as plants have been exposed to increasing weeks of cold, you get this progressive switch with a higher proportion of cells not expressing FLC anymore. Because if you grind these up, it looks beautifully quantitative, but it's not average at each gene, it's a population average of a switch. Now this mosaic type effect of FLC silencing is then averaged because the downstream target of FLC is a gene called FT, and the FT protein is mobile through the plant, and this averages the FLC mosaicism. So over many years now, we've been studying this process, and I've got one slide here just to give you the overview of all of our current understanding before I mine into one aspect. So as the plants germinate, FLC expression is established. That involves a series of antisense transcripts, which are very complex. They're differentially RNA processed, and depending on the level of expression, the plant then has an overwintering phenotype or can actually cycle without winter at all, like you have a rapid cycling phenotype. But as the plants, these antisense transcripts then become very important as the plants feel the cold, in that their expression goes up very dramatically. That leads to a shutdown of the sense expression. And at later phase in the cold, you have a much slower induction of a protein, which I'll talk a lot about, VIN3. It's PHD protein, which is an accessory protein to polycomb repressive complex 2, which is this very conserved chromatin complex that delivers these K27 trimethylation modifications. As the plants then come back in warm, this nucleated region spreads across the whole locus, and you have long-term silencing involving K27 over the whole locus and classic polycomb memory until you get the seed formed, and then the expression is reset. So there's many papers, I've looked at many different aspects, but today what I'm going to talk about is really thinking about that question I posed at the beginning. How are plants registering noisy temperatures, and how do they distinguish prior exposure? So I'm going to focus on this cold step. So as I said to you, that a cold step involves induction of these transcripts antisense to FLC. They initiate pretty much at the sense polyase site. They're very strongly upregulated in the cold, and somehow these fluctuating temperatures are important. But what was clear when we started to look at the single molecule fish, so green shows you the nascent antisense transcript, and red the sense transcript on Arabidopsis nuclei. I show a subset of the data here, obviously, but what you can see is that at each locus, and you're looking at the Arabidopsis gene in the nucleus here, you only get either red or green. So at each moment, there's only antisense or sense transcribed mutually exclusively. And so if you pulse antisense transcription, that's a great way of shutting off sense transcription. So part of the shutdown in the cold is induction of these transcripts, these antisense transcripts, which we christened couleur. Now, these are important in cold, but some recent data from some field experiments, which we were extremely fortunate to be able to do in collaboration with 2 groups in Sweden. We had some fantastic MSc students who went out very diligently every week or so, harvested the plants that we'd sown in the field and sent them back to Norwich so we could do molecular analysis over a whole season. So here's the temperature logger next to the plants. And here's now our analysis where we're looking at these antisense transcripts. And suddenly, as we did this experiment, there was one day with this huge peak of antisense transcription. And we were like, well, what's happening on this particular day? And then if you look at the temperature profile, it was the first night the temperatures dipped just a tiny bit below 0. Now we've managed to get chambers in Norwich that recreate those temperature conditions. And yes, indeed, it's the just below 0, which super induces the couleur transcription. Does that have an effect on FLC? Yes, indeed. When you look at the FLC RNA in response now to actually very different fluctuations. So these are cabinets where you can fluctuate the temperature, but not actually go below 0. And you can fluctuate the temperature with a mean still of 5 degrees, but now go below 0. You see a very strong effect of these freezing temperatures on sense transcription. So we have acute freezing temperatures being registered. But this second step that I want to talk to you about, which is accumulation of this accessory protein to PRC2, VIN3, is a very different mechanism. Again, we went into this through the genetics. Somehow this is slow, and that explained the slow accumulation of the K27. But when we looked into the mechanism of how cold was inducing VIN3, it was a totally indirect mechanism. Cold itself doesn't induce NTLA. This shows that this is the regulator of VIN3 that was important. But what happens is that the protein, this direct regulator of VIN3, is a very stable protein. And because of reduced cell division in the cold, this regulator of VIN3 accumulates because of reduced dilution due to slow cell division at low temperatures. So here we have a temperature-dependent growth contributing to long-term cold sensing, quite a different mechanism to what was expected. So in fact, when we look at this array of temperatures that the plant is trying to register, what we found is that we're looking at the plant's developed mechanisms to monitor both acute, as I showed you, the cool air induction upon freezing, but also these long-term through these indirect growth mechanisms affecting the accumulation of proteins which directly regulate this accessory protein to PRC2. But through some data that I haven't been able to tell you about, every time in the afternoon it goes above 15, then all of the VIN3 that's accumulated gets turned over. So the plants are also monitoring peak afternoon temperatures to register, is this really winter or am I still in autumn? And just you can see from this profile, autumn is often characterized, even with cold nights, by high temperatures in the afternoon. And this was not something people had thought of before. It was always vernalization was cold as opposed to absence of warm. So what we've got now are different thermosensory steps, but as we mine into this more and more and more, we realized that every step we looked at in our mechanism was thermosensitive, and that we were getting integration through accumulation of VIN3 with this long-term cold, the short-term absence of warmth, daily current temperature. Also, the nucleation step was temperature dependent. So there was many temperature inputs. Of course, this is biochemistry. Temperature will affect biochemistry, but different aspects of the temperature profile were feeding into different parts of the mechanism. And that instead of one or two major thermosensors, we had distributed thermosensing, leading to now nucleation of FLC. So this nucleation site, what do I mean by that? Well, again, genetics came to our rescue. This is nucleation of this polycomb complex at a site actually within the first exon, first intron of the FLC, identified by a single mutation, which took us to looking at a particular DNA binding protein, happens to be called VAL1, which interacts with a range of proteins, including some complexes involved in RNA processing and turnover. Now, this was quite nice in that it linked a sequence-specific DNA binding to the chromatin, the polycomb complex. That would suggest, as many people consider, that a lot of this chromatin regulation is trans-targeted by the VAL1 trans factor. But it wasn't as simple as that, because when we analyzed nucleation, and this was done in a mutant where we could just isolate nucleation and prevent spreading across the locus, in a system, again, looking at roots where we looked at two transgenes, now FLC fused to Venus and FLC fused to M. cherry in the same nucleus. What became clear was that when we got nucleation, it was a digital effect, which had a manifestation on the chromatin. You switched from K27, K36 to K27 trimethylation. It was then epigenetically stable. Once it nucleated, which was a very low probability of nucleation, but once it nucleated, it was metastably stable for about 10 cell cycles. But that happened independently at each transgene. So there was something local, something in cis driving the nucleation. So the current view at the moment is that we've got multivalency between this collection of proteins, which are nucleating this silent state, and that is capturing all of this temperature complexity, and with a very low probability, capturing and then preventing that allele from being expressed again and buffering it from any further temperature. Now, an interesting aspect of that is that, as I said to you, there was something local. I also said that once you've nucleated, you're now epigenetically silent. So this is inherited to daughter strands after DNA replication. But how can this be? And actually, in this theory paper, which has just come out, and I haven't got time to go into all of this, but the argument is in the polycomb field that polycomb is inherited through what's so-called read-write mechanism, where the chromatin complexes bind to the K27 trimethylation and fill in gaps. But you have to have a certain size K27 trimethylation patch for that to be robustly inherited. And this patch of 3 is just not enough. So that's taken us into a very interesting collaboration just recently, looking at the properties of these proteins now, these proteins that come up with PRC2, these 2 proteins, VIN3, which is this cold-induced one, and actually a homologue of VIN3 called VIN5, which is constitutively expressed. And it's a very exciting collaboration that we have with Marianne Biensis' lab at LNB, Mark Fiedler and Elsa Franco Echevarria, where we've looked at the properties of VEL proteins to ask, how can it be nucleating in a sort of switch-like digital mechanism? And how can it then be stably inherited through DNA replication? Now, this is the structure of those proteins. There's some domains at the amino terminus where it looks like it might be involved in chromatin association. But at the C terminus, there was a domain that we termed many years ago, the VEL domain. It was clearly important for homo and heteropolymerization of the VEL proteins with each other. And in some recent unpublished work, now Marianne's group has shown that this VEL domain is a new polymerization module. We've defined now the interfaces, the amino acids that are important for the interfaces. It's a 4 alpha helix bundle, which folds to form these Lego blocks, is the way I like to see them. They form a sort of molecular Velcro. They form large assemblies in vitro. One of them actually forms cross-links as well. In vivo, you see that they're giving these dynamic puncta, which are interface dependent. When you mutate the interfaces, you lose the puncta. So these proteins are conferring properties which potentially are extremely important for this digital nucleation and then the epigenetic maintenance of that silent state through replication. So let me just summarize where I think we are. We've got the questions that in our minds that we were looking at. How are plants monitoring this complex temperature and then determining if it's had this long-term period of cold? So all of the cells, and I show you here just the roots, but remember this is a schematic, it's probably going to be true for all of the cells in the plant. When they germinate, FLC is on. There's a color coding green here. All of them have got FLC in on state. Slowly but surely, in response to these different temperature signals, both long-term and acute, a low probability switch occurs. And that is low probability. So slowly but surely, during weeks of cold exposure, more and more alleles switch. And I sort of see this as a bit like a sand clock, that you've got the grains, one chamber, and they're all green, and then slowly but surely, each allele, which would be a sand grain, transferred to the bottom chamber. That's then the epigenetic switch. Now, when I draw these, I've just shown here the meristematic cells in red. Probably all of the cells nucleate, but it's the meristematic cells which are really important for the next phase of the plant, because as the plants grow out in the warm, the meristematic cells make up the body of the plant. And so after long exposure, then all of the tissue is got FLC silencing and the flower bake is fully released. So let me wrap up. The question we started, which was this very plant-specific protein, how are plants registering winter and knowing spring has happened to align their flowering very favorable seasons of spring. And we've ended up looking at this very conserved epigenetic switching mechanism. Now for Arabidopsis and brassicas, FLC is the important target. I think as we go forward, what we'll find is that in all plants, there are somewhat similar mechanisms just acting on different targets. What we found is instead of, as was thought of, perhaps 1 or 2 major thermosensors accumulating with different temperature time dynamics, what we've got is a distributed temperature sensing network. That this prior temperature exposure is through this sand clock type mechanism, where slowly but surely a switch occurs and then that stays off, and that's the memory. That this epigenetic switch involves protein polymerization. I think this is going to be a mechanism which is widely applicable in many chromatin mechanisms for conferring epigenetic memory. And through some work that I haven't had a chance to talk about, when you then look at adaptation of Arabidopsis to these very different winters that it's exposed to, Arabidopsis grows from the Arctic Circle almost to the equator. What we found is that inevitably, because we're dealing with a network of very conserved trans factors, polycomb transcription factors, which have thousands of targets in the Arabidopsis genome, that all of the adaptation is actually through non-coding cis-localized SNPs, which influence how these proteins are talking to the locus. So that leads me to just thank the people who've been involved in the work. This is a long-term collaboration with a theoretical biologist, Martin Howard, and his postdocs, who are the ones labeled in red here. Again, a very exciting recent collaboration with Marianne Bience's lab and Mark Fiedler and Elsa Franco Echevarria. And then the individuals in my lab, this is rather an old photo, as you can tell, we're all standing next to each other, are the ones in white with our new arrivals on the right here. Thank you for your attention.
Thank you very much indeed, Caroline. A wonderful illustration of the mechanistic insights that come from looking at plants in their real environment and not just in growth chambers, and trying to understand how they're adapted to real environments. Right, so our next speaker is Luis Herrera-Estrella. Known Luis many years, nearly 40 years, and because he's been a real leader in investigating agrobacterium biology and inventing ways of enabling us to use agrobacterium with plant genetic manipulation. And he's gone on to make a number of interesting investigations into plant biology. He's a emeritus professor at Sainsbury, and he's a professor at Texas Tech University. And he's going to tell us about the regulatory network controlling seed desiccation tolerance, that's reactivation in vegetative tissue, because this is a process that plants have to go through between being formed as seed to tolerate that desiccation, and then to absorb water and grow subsequently. So Luis, looking forward to hearing your talk.
So thank you very much for the kind introduction and the invitation to participate at this very interesting seminar. And so I'm going to talk about a very fascinating, oops, I cannot, something happened. A very fascinating process that is desiccation tolerance. So desiccation is the capacity of an organism to survive in the dry state for long periods of time. So we have, for instance, the example of Artemia salina, which eggs are viable for over 10 years in the desiccated state, and the little embryos that are in the eggs survive the dry state during this time. And there is also these little animals, the tardigrades or water bears, that are extremely resilient to environmental stress. They can survive 50 years in the desiccated state. And when you water them again, they come back to life. These little animals are also very resilient to radiation. They have been taken to the space station and exposed to cosmic rays, and they can survive very well this very harsh environment. In plants, desiccation tolerance has been a very important process. I had a video here which I cannot play anymore. I don't know why. Oops. So early land plants had to acquire desiccation tolerance to be able to survive. Once they started to move away from the edge of the water, they had to tolerate longer and longer periods of dryness. So they had to evolve mechanisms to survive in the dry state. This is probably inherited from the algae from which early land plants evolved from. So this desiccation tolerance evolved, but perhaps the most amazing process of desiccation tolerance is desiccation tolerance in seeds. So here I have a few examples. So these are, for instance, acacia seeds that were rescued from an herbarium in Iran by a Swedish scientist. And when you take these seeds that are 150 years old and you put them in media, they still are viable and germinate. There are also some other even more striking examples. Lotus seeds from a 1300-year-old thumb in China, in which these lotus seeds were again still viable. And the record so far is 2,000 years old date seeds that were rescued from an Egyptian thumb. And when you again, you saw them in a proper soil with water, they are still viable. So that means that during seed development, plants have to activate a very complex system to protect all the cellular structures, membrane structures, ribosome, DNA and RNA integrity, chloroplast membranes, so that they can survive in the dry state for all these periods of time. So when plants evolve, early plants didn't have proper root systems, and they didn't have vascular systems, so they had to develop desiccation tolerance in the vegetative tissue so that they could survive to the periods of dryness. Later on, plants developed true roots, vascular systems, and they no longer require the capacity to be vegetative tolerant to desiccation. Vegetative tolerant desiccation that was very important in the early branches of plants was lost when tracheophytes evolved. So when the vascular system evolved, the plant had the way to extract water more efficiently from the soil, so they didn't require anymore to have vegetative desiccation, so the vegetative desiccation was lost. And it has revolved many times during the evolution of land plants, particularly in the angiosperms, in which a vegetative desiccation has revolved at least 13 times. So if we have a system in which vegetative desiccation is present in the early land plants, then it's lost and hijacked to be rewired to be expressed in seeds. And this vegetative tolerance in seeds is then rewired again to be back in the vegetative tissues, suggests that although this is a very complex process that requires expression of late embryogenesis, abundant proteins, heat shock proteins, elite proteins, a mechanism to protect against oxidative stress, reactive oxygen species. So how can all these very complex mechanisms be activated and rewired to be activated in different tissues? So that's one of the questions that we would like to understand and test whether we can actually reconstruct an event of plant evolution. If you have desiccation tolerance in the seed, can we reactivate that pathway in vegetative tissues? So during the process of seed development, the early steps in seed development is mainly cell division and cell differentiation, in which the embryo proper is developed. Then, after a time when you have an almost fully developed embryo, the seed starts to lose water and it starts to accumulate storage compounds, lipids and carbohydrates. At some point during this process, when water is lost, the program to survive in the desiccated state is activated. So all these genes that are required to protect the cell structures to maintain viability is activated. So how can we understand this process? So fortunately, many people for a long time started studying embryo-lethal mutations. The idea was to identify genes that were required for embryo formation and embryo viability. Some of these mutants were actually not embryo-lethal, but they were desiccation intolerant. So if you could rescue the seed before it dried out, the embryo was viable. If the seed desiccated, then the embryo was dead. So these mutants are very interesting because they are mutations in master transcription factor regulators that regulate the transition from the embryo development to vegetative development. So in these mutants, the embryo doesn't go to the desiccation state and dormancy, but they rather directly germinate after embryo formation. And this is seen because you get trichomes in the cotyledons, and they remain green rather than becoming white. So there are mutants in these transcription factors which are desiccation sensitive and desiccation tolerant. So they have a very similar phenotype in the transition from embryo to vegetative state, but some lose desiccation tolerance and others don't. So if we compare these mutants, then we can start identifying the genes and the program activated for desiccation tolerance. For instance, the LEG1 mutant and the LEG2 mutants are very similar. They both accumulate chlorophyll in the dry seed. They accumulate anthocyanins in the cotyledon. They have reduced storage compounds. They produce ectopic trichomes, but one is desiccation tolerant and the other is desiccation intolerant. So if we compare the global transcription in these two seeds during seed development, we should be able to evaluate identify all the genes that are activated to acquire desiccation tolerance. There are other mutants, for instance, AB35 is a mutant which is no longer desiccation tolerant, but there are alleles of AB31 which are still desiccation tolerant, although they have still reduced ABA sensitivity. So we made comparisons of these mutants at 3 different developmental stages during seed formation. So we analyzed RNA-seq data and we analyzed the metabolomic profile of these seeds during the development. So the idea was to see whether we could identify the set of genes that are required for acquiring desiccation tolerance. So we found 2 major sets of genes, those that are activated in these mutants that are intolerant to desiccation. These genes have to go these genes have to do with the transition from embryonic to vegetative growth, because they encode genes involving chlorophyll biosynthesis, antenna formation. And there is 2 other sets of genes which are not activated in the intolerant mutants, but they are activated in the tolerant mutants. These sets of genes are the ones that we believe are responsible for the mechanism. So in the genes that are upregulated in the intolerant mutants, of course, we have photosynthesis, sucrosynthesis, cell division, and cell expansion, because these embryos go to the vegetative growth. The genes that are not activated in the intolerant mutants have to do with lipid biosynthesis, storage proteins, and straight related responses, LIA proteins, oligosaccharide biosynthesis, heat shock proteins, antioxidant mechanisms, and so on. So to understand how this is activated, we build a regulatory co-expression network of seed development. So we use about 350 sets of gene expression of seeds in different processes, different mutants. So we build this regulatory network. And in this network, we identify all the genes that are activated, all the transcription factors that are activated during seed formation. And among these transcription factors, we identify those that fail to activate in the mutants that are no longer desiccation tolerant. So in the expectation that these transcription factors are the ones that orchestrate the activation of the desiccation tolerant mechanism. So we found 2 subnetworks that are active, that are not activated in tolerant mutants, but are fully activated in the tolerant mutants. These 2 subnetworks have different properties. One of them has a dual role in activating the expression of genes involved in the accumulation of reserve compounds, and also in genes that have to do with the stress tolerance, for instance, the accumulation of LIA proteins. So we can see here in this network, for instance, all these yellow dots are LIA proteins. Some of these other proteins are heat shock. Some of them have to do with the accumulation of sucrose, and and oligosaccharides that are required for desiccation tolerance. The second subnetwork is fully involved in activating cell protection mechanisms. Again, it regulates the accumulation of many LIA proteins. It also accumulates proteins that are involved in stress-related processes, synthesis of oligosaccharides, heat shock proteins, and so on. These two subnetworks change during the evolution of seed development. So the subnetwork alters its architecture during seed development. First, initially, it's only involved in the accumulation of reserve compounds. Then it starts to activate genes involved in stress tolerance until more or less half of the network is part of stress tolerance and the other half is involved in reserve compounds. Here it's important to mention that at 17 days is when the accumulation of sugars and other compounds are activated during the desiccation tolerance mechanism. The second network is only activated when water is reduced enough in the seed to activate the desiccation process. And this subnetwork also evolves and activates many genes involved in desiccation tolerance. So if these networks are real networks, mutations in the transcription factors that control these subnetworks should lose seed viability during desiccation. So we produce mutants in different transcription factors of these subnetworks. We produce seeds and we evaluated the seed viability after 30 days of desiccation. So as you can see, the wild type has almost 100% of germination, and the different mutants have different rates of survival after desiccation, suggesting that in these transcription factors have to do with desiccation tolerance. If it is a network, double and triple mutants should have further reduced their tolerance to desiccation. And that's the case if we have a double mutant or a triple mutant, then the seed becomes almost desiccation intolerant. To confirm that these transcription factors had to do with desiccation tolerance, we ectopically express some of these transcription factors in the AB35 genetic background. AB35 is completely desiccation intolerant. And we can see that the ectopic expression of some of these transcription factors partially rescue the capacity of the seed to survive short periods of desiccation, suggesting that indeed these networks exist and these nodes, which are transcription factors, are indeed coordinating the expression of a large number of genes involved in desiccation tolerance. So what we want to do is to reconstruct this event in vitro. What happens if we are reactivate in vegetative tissues the expression of these networks? So to assay for this desiccation tolerance, what we do is we germinate the seeds for 14 days. After 40 days, we have 10 days of full irrigation, and then we stop watering for different periods of time. It could be 2 weeks, 3 weeks, 4 weeks. And then we do a recovery irrigation treatment, and we count the plants that actually survive the period of dryness and how many of them die. So we have done these experiments using the expression of different combinations of 2 or 3 different transcription factors. And you can see that the survival of the plants to 30 days of desiccation increases when we express several genes. And when we reach a whole one of the subnetworks, when we express the 5 most important nodes of subnetwork 2, we almost reach 100% of survival after 30 days of desiccation. This is the plants, how they behave. This is the control in which we stop watering. We did the recovery irrigation, and the plant, all of them die. These are expressing 5 transcription factors. We desiccate them. we water, we do a water recovery, and they all survive. Here it is interesting to note that if you constitutively express these transcription factors, they reduce the growth of the plant because probably constitutively activating the expression of LIA proteins, heat shock proteins, the accumulation of oligosaccharides required for desiccation tolerance has a big penalty in growth. If we induce these genes by an inducible, drought-inducible system using promoters that are responsive to drought, then we can see that the plants grow normal, but they are still fully resistant to this process of desiccation. These plants are still not yet 100% desiccation tolerant. 6 months without water, they die. So, but we are now trying to activate the two subnetworks using the CRISPR-Cas system for gene activation to see if we can orchestrate the activation of two sets of subnetworks and see whether we can actually reconstruct this evolutive event in nature. Of course, activating these genes also provides some information about how to design plants that are drought tolerant. So depending on the combinations of transcription factors we activate, we get different levels and different mechanisms of drought tolerance that might be useful for the future breeding of drought tolerant plants. And this is just my research group, and the people working on this topic is Sandra Gonzalez, Telma Rico, and Gerardo Alejo. And I thank you for your attention. I'm sorry that my video didn't run properly. I don't know what happened. Thank you very much.
Luis, that was a fantastic, lovely illustration of RNA-seq-based discovery and insight into mechanism. So I think at this stage, as I understand it, we're going to have a discussion between the speakers. So I'd like to encourage you all just to think about what you might have learned, sort of a higher level insights into mechanism and diversity, and who wants to kick off?
I have a question for Sandra.
Please.
Sandra, you mentioned that most of the domesticated plants cluster in a specific region of this global spectrum of plant diversity. Is that a coincidence, or is that these plants have some traits that facilitate their domestication or their capacity to produce food?
My impression is that it's not a coincidence that they were being first selected because they originally in the wild types originally had some desirable traits, and they were then further selected towards desirable traits. So for example, the fact that the plants are relatively small, the plants, more importantly, rather than being small, they grow fast. Most of the plants we grow, most of the plants on that corner of the spectrum grow very, very fast, and they have very palatable and very acquisitive leaves, but the leaves don't last very much. And they do have nitrogen in somewhere, either nitrogen in the leaves, which made them very nutritious to our animals, and they have all these alkaloids, which we use for various recreational or medicinal or whatever use. And they are easy to grow during early agriculture, right? You just plant the seeds and you have plenty of germination. So those were the traits first filtered from the wild. And some of them are wild, some of them are weeds. All the agricultural weeds are in that corner too. And then, of course, we perfected the traits we wanted. We made the seeds bigger. So that's a corner in which our sculpting of phenotypes has been strong. Actually, the only corner in which it has been strong.
This is very interesting because there is a lot of discussion that we have limited our food production to a very small percentage of plant diversity, and that we should expand to use many more plant species for food production. So do you think that's feasible, or these tell us that biologically we have already selected the best candidates?
No, I think that if you remember, I showed 2 hotspots. There is one hotspot which are all herbaceous plants, and then there is another hotspot which are mid-sized trees with big seeds. That hotspot has already been tapped because those are trees that don't grow too slowly and they produce big fruit. The body of the plant itself is not very practical to us. It's not very palatable. It takes a long time to grow, but then they have these really nutritious fruits. And most of the tropical fruit trees are in that particular spot. Now, the other areas of the spectrum, the problem they have is that they don't grow fast. So it's probably not very practical to us. They are good for standing incredibly hard environments. So if we are interested in taking a page from nature on how you get a plant which is incredibly adapted to a tough environment, then we should be exploring the southern and southeastern corners of the spectrum.
Right.
I was going to slightly change the discussion, because what was interesting to me, Luis, is that you approach the desiccation with the RNA-seq and the network analysis. And a lot of people are doing that now for flowering, because obviously if they want to do it in different plant species, rather than go through all the full genetics, they tend to do RNA-seq. And actually the RNA, the networks that come out, you can see the same regulators that we've discovered through the forward genetics, but I'm not sure you would have necessarily picked out the major nodes until you've done the natural variation analysis. And it's the natural variation, it's the evolution's tweaked the various important nodes. That's how we discovered FLC and major regulators of FLC rather than. So I wondered, have people done natural variation of desiccation tolerance and then mapped that onto your networks?
We are currently working with resurrection plants, doing a similar analysis comparing species that are naturally desiccation tolerant versus closely related species that are intolerant, and also with grasses, and to compare their responses to naturally desiccation tolerant and intolerant grasses, and building the networks to see how they change during the evolution of plants, because that obviously is going to lead us to the central regulators.
Well, I think the intra-specific analysis may be faster, where you can actually do the cross and the genetics and the QTLs, as opposed to, you know, just a lot of network analysis. Yes, we are doing also the crosses. It's not that easy to cross a Laginella, which is our best model to do this. But we are also trying to generate mutants that become now intolerant. So, but the thing is that the response of all plants is very similar. So the effector genes is the same family of genes that are involved. But yes, indeed, we still need to go backwards to find the central regulators, which actually might be these developmental regulators like LEG1, because LEG1 controls the 2 processes, both the transition from embryo to vegetative growth, which it suppresses, and the activation of desiccation tolerance. So what we think is that we are just below LEG1 in the activation of desiccation tolerance.
Great. Can I just, before we have more discussion, I just want to say to the audience, pretty soon we'll be switching to audience questions. And so far we've had 2, but we really love more. So I really encourage audience members to type their questions into the Q&A bar. Sandra.
Yeah, a question to Luis. It's a very at the macro level. You show this phylogenetic tree, so you obviously look at the bigger picture. Is desiccation tolerance cluster in any particular plant groups today? in terms of
no.
phylogeneity or in terms of growth forms?
No. There are dicotyledonous plants that are desiccation tolerant. There are monocotyledonous plants. There are many, it's distributed among many different plant species. So it's not that it's clustered and it generated many desiccation tolerant plants. That's why we say it has re-evolved independently 13 times, because they are in different branches of plant development.
So just on that, so I think Criteria-stigma, I think, is the classic resurrection plant. And I mean, have you looked at, there's a lot of expression data, I'm sure, available from Dorothea Bartels and people like that. So can you see the same kind of profile showing up during desiccation of Criteria-stigma that you see during the time course you looked at in Arabidopsis seeds? And I think you can transform Criteria-stigma. You know, could you go in and mutate the orthologs of the genes you've identified and see if that leaf tolerance of desiccation goes away, or if there's been maybe even duplication of genes so that there's some for seed desiccation and some for whole plant desiccation tolerance?
Yes, we have analyzed the Criteria-stigma and the Selaginella expression data, and the networks are similar, but they are not identical. So there are some common elements, and some others are different transcription factors that belong to the same family, but change from one species to another. They are not direct orthologs. And in some cases, we get the incorporation of completely different transcription factors as nodes. So the rewiring, although seems to have a general trend, they are not identical in all cases. So that's something we are working with, and we have also developed the transformation of Selaginella. So we want to do the knockout of these genes in Selaginella.
Great. I've got a question to Sandra, actually. So in terms of biodiversity and human impact, something that we've done over the last 150 years is to elevate CO2 levels. So you can imagine that the competitive advantage of C4 plants with a more efficient photosynthesis, when it's 280 ppm, is going to be a lot less than at 420 ppm or whatever we are now. So I guess the first question would be, if you classify plants by C3 and C4 plants within those principal component analyses you displayed, do you see a pattern? Is any evidence for that pattern changing as CO2 levels go up?
I'll answer in two bits. First, there's no much clustering of C4 on the spectrum. It is slightly more common in the, say, the western part of the spectrum, simply because C4 is very common in grasses and other groups. It's not homogeneously distributed among families. And so it is there, but it is not associated. So having said that, C4, for example, if you look in grasses, you have C4 grasses, annual acquisitive, C4 grasses, extremely conservative, C4 grasses, very tall, very small. So it's kind of orthogonal, I would say, almost to this global spectrum. Okay,
thank you very much. I think time's pressed on, and we've overrun slightly on our intended time course. There's a few interesting questions from the Q&A. So Richie Perrick asks, I presume this is for Caroline, where are these signals detected? So is the detection of temperature equally in the leaves, in the shoot meristems, or in the roots? What do you know about where the detection happens?
The detection is in all over the plant, basically. That was the sort of picture I was trying to convey at the end, that all the cells are independently detecting and remembering those signals. And then it's combined into this sort of integrated effect through this mobile signal FT. So it's not, And this is very different from what people were expecting. There was an expectation it would be in the meristems or very specific parts of the plant, but certainly it's all of the, you know, the major growing parts, which is really the bulk of a small plant, is all detecting the temperature.
Thanks. So there's a question from Rob Martinson. I think I'll put it to Sandra first. So what do you think about perennialism, and where do perennials lie on this principal component analysis map that you showed us? And you tell us, well, I guess the second part of the question is to Caroline, on what happens, how do flowering signals work in perennials? First, Sandra.
Sorry.
Perennials, do perennials and annuals map very differently in your...
Oh yes, sorry. Yes, definitely,
space?
Definitely. All the annuals are on the west. Yes, it's on the west, and it could be anywhere in the west. It could be northwest or southwest, but all of them are there. These are all the herbaceous and small plants, whereas there's zero annuals on the east of the spectrum.
Right.
Yeah.
So what about
my input onto perennials? So George Coupland's lab has been looking specifically at the basis of perennials in an Arabidopsis relative and has come down to FLC regulation again. And it turns out, I think the simple answer would be, it's the SNPs within the gene itself, non-coding SNPs, that prevent the switch to the epigenetic silent state in that long-term memory. So there's a kind of slightly longer than metastable, but not long-term. And so the gene is reactivated always in the spring. And so some parts of the plant, which are old enough, basically when FLC shuts off in the winter, the positive signals actually start being expressed. Those are then epigenetically on. So even when FLC comes back on, there's still that positive signal. But in other parts of the plant, because of an age-related pathway, those positive signals can't come on. So there's a beautiful combination between non-coding SNPs and FLC and an age requirement for vernalization to happen. And that can then explain the perenniality.
Can I just say something? Isn't the case that many annual plants have relatives that are perennial? For instance, sorghum has perennial relatives, yes. Maize has perennial relatives.
Yes, I think annualism has arisen from perenniality many times. And certainly in our data set, the annuals will have arisen from perennials.
No, but I mean in the map, if they separate completely in the West, there must be many perennials.
Oh, many. In the West, there are many perennials. There are acquisitive, short-lived, small perennials, yes. So the perennials can be everywhere, but the annuals are only in the West. And I found the same as Caroline. Many annuals are rare members of mostly perennial families, with a couple of exceptions.
Okay, so that's the direction of evolution.
Yeah.
So I've got a question here for Luis. So could you compare desiccation tolerance to salinity tolerance, and could the approach you've taken to investigating desiccation tolerance also be applied to investigating salinity tolerance?
The response in the general response is similar in terms of the type of effector genes that are involved in salt tolerance. And some of the transcription factors that have been reported for salt tolerance are also part of the families or the same we find in this network. But I don't know if the network analysis has been completed to really know how similar the networks are. But in general, when you increase drought tolerance and desiccation tolerance, you get higher salinity tolerance. So the mechanism must be common in many components.
Another question for you, Luis. What about cacti? Do they actually have desiccation tolerance, or do their adaptation more to avoid exposure to desiccation?
Yeah, the cacti and many other desert plants, they have mechanisms of avoidance of desiccation. They store water, they have a different CO2 assimilation pathway. So they function in a different way to preserve and accumulate water to withstand dryness. It's not that they are desiccation tolerant.
Another question here from Jason Lynn. I think it seems to be a question about quantitative efficacy of your engineering desiccation tolerance compared to, let's say, let's call it the extreme desiccation tolerance of a Cratera stigma. So how far are you away from, engineering the transcription factors of Arabidopsis such that you could dry them out and completely and then revive them the way you can with the resurrection plant?
Well, we are probably 6 months away of activating all of them and see how far we can go in desiccation tolerance. As I mentioned, activation of 5 gives a very strong drought tolerance, but it doesn't confer complete desiccation tolerance, because if you keep this plant dry for a long time, they die. So we'll see in the next probably 6 to 12 months, I should know how far we can go in quantitative terms of desiccation tolerance. Okay,
got a question here to Caroline. I hope I can do justice to it. I'll just read it out. Since cold exposure memory is occurring on a population scale, is the robustness or accuracy of the vernalization decision dependent on the size, i.e. number of cells in the root structures? And thanks for a great talk.
Thank you, Nicole. I think, my schematics inevitably had to be rather simple. So it's not just those few cells that are informing, it's a much broader set of cells. So I think it's more the probability of that switch then determines the proportion of the plant that is silenced FLC or not. And that is changed in natural variation. So that, the SNPs at FLC do change that. The sand clock model, the aperture would be different depending on whether it's adapted to Sweden or France or wherever. But I think the number of cells rather than the size or the size of the meristem or anything like that, because it's a very large proportion of the plant that is silenced.
Fantastic. Well, I think we've reached 3:30 now. So Janine, I think we're about to switch to the phase with 3:30 in England and 10:30 where you are. The phase where we have the early career scientists meeting the panelists.
Yes, indeed. We have reached that time. So before we move on to the Meet the Panelists session, I just want to thank all 4 of you, Sandra, Luis, Caroline, and Jonathan, for really great talks and a really nice discussion today. Thank you so much for being with us. Yes, trainees, please stay on to meet the panelists and hear from them about various career-related and mentoring-related topics. To all of the audience, thanks so much for joining us today. Come back on November 3rd at 9 a.m. Eastern Time for our next Life Science Across the Globe, which is focused on connectomes and is being hosted by the MRC Laboratory for Molecular Biology. So I hope you can all join us then. And yeah, so trainees, please stay on the call. We'll take 30 seconds or so to let everybody depart who wants to depart, and everyone gets situated. Please go ahead and complete the survey. The link is in the chat box. Your feedback is super important to us. Thanks, everyone.